This video is about post-translational protein modification, shortly represented by PTPM, post-translational protein modification. The nascent polypeptide formed at the end of genetic translation is not a functional product, so it cannot serve as a mature functionally active protein. To become a mature functional protein, it has to undergo structural modifications, functional maturation, and chemical processing. This may be called post-translational modification of protein. Essentially, it involves the covalent bonding and enzymatic modification of inactive nascent protein. PTPMS or post-translational protein modifications include primary and secondary modifications. Primary modifications are mainly structural modifications and they include protein folding and proteolytic cleavage. Secondary modifications are mainly chemical modifications. They include the covalent incorporation of chemical grouping at the N-terminal, C-terminal, or the side chains of some amino acid residues, with the exception of alanine, glycine, leucine, isoleucine, methionine, and valine. Glycosylation, methylation, acetylation, alkylation, and phosphorylation, along with that hypoxylation, etc., are the common methods of chemical modifications. The primary and secondary modifications may take place even during peptide chain elongation and also after translation termination to attain the characteristic form and specific functions of proteins. In some cases, several polypeptides get assembled to form a complex functional protein. First, protein folding. Protein folding is the characteristic folding of a nascent linear polypeptide chain to attain the unique secondary or tertiary three-dimensional native structure of the functional protein molecule. It involves the interaction of the amino acid residues of the polypeptide chain. As a rule, protein folding is a cooperative process. This means that each folding induces amino acid interactions to initiate the next folding. The characteristic folding and assembling of polypeptides to assume their 3D native configuration are mediated by some catalytic proteins called chaperones. They bind with nascent polypeptide and prevent their incorrect folding and wrong assembling. Chaperones and folding of polypeptides. Chaperones, also called molecular chaperones, are the specialized proteins which enable individual polypeptide chains to precisely fold into their final thermodynamically stable functional configuration. They are also involved in the insertion of intrinsic proteins into the lipid bilayer of biological membranes, the transmembrane transport of proteins, the correct refolding of denatured proteins, etc. So, chaperones play a significant role in correct folding and spatial assemblage of polypeptide chains to form the three-dimensional tertiary structure of functional proteins. At the same time, they are not themselves part of the functional complex of proteins and they do not directly provide the direct three-dimensional structure of proteins. They simply bind to nascent polypeptide chains and allow them to fold only in the correct direction, preventing their misfolding. Abnormal folding of protein in human beings without regulation of chaperones often leads to Alzheimer's disease. The catalytic activity of chaperones is energy dependent and so it involves ATP hydrolysis. Then proteolytic cleavage, proteolysis. Next step. In a broad sense, proteolysis is the degradation of the peptide chain by the breakdown of peptide bonds. In the post-transcriptional modification of proteins, it involves four main events, namely cleaving out of methionine, removal of signal peptide, trimming of terminal amino acid, and removal of internal peptides. Proteolytic cleavage is mostly associated with the activation of inactive precursor proteins. Methionine is the first incorporated amino acid of polypeptide chain. Once the amino terminal of growing peptide chain is established, the initial methionine is often removed from the amino terminal. Signal peptide or signal sequence is a linear series of amino acids at the N-terminal of a nascent polypeptide chain. During post-translational modification, it is cleaved out by the enzyme signal peptidase. present in ER, endoplasmic reticulum. Then the processed peptide chain is released for packing in Golgi bodies and lysosomes. Sometimes peptides may be subjected to a process of breaking and making of covalent bonds. This is catalyzed by enzyme disulfide isomerase and peptidyl propyl isomerase. Then some major aspects related to chemical modification. The chemical modification of protein is accomplished by several chemical processing methods. Some of them are mentioned below. First, glycosylation. Protein glycosylation is the incorporation of mostly oligosaccharides or glycan to the polypeptide chain catalyzed by the enzyme glycosylase. It is a major process of post-translational protein modification. It has significant effects on the folding, molecular conformation, stability, and functional activity of proteins. Even then, only very few proteins are glycosylated. In protein glycosylation, the glycan is added either to a nitrogen atom or to an oxygen atom. Thus, there are two kinds of protein glycosylation, N-linked and O-linked. N-linked and O-linked. The former usually occurs on the amide nitrogen as a paragyne, while the later occurs on the oxygen atom of serine or threonine. The N-linked or O-linked oligosaccharides have significant roles in protein sorting, immune recognition, receptor binding, inflammation, and pathogenicity. For example, the N-linked glycans of an immune cell can dictate how the cell has to migrate to specific sites and how it can recognize the self and the non-self. Then protein methylation. This is the addition of methyl group usually to the lysine or arginine residues of the peptide chain catalyzed by an enzyme methyl transferase. Arginine can be methylated once or twice, while lysine can be methylated up to 3 times. Protein methylation is significant in the following aspects. Provide the protein stability and protection against enzymatic degradation. Exert significant effect on protein-protein interaction, protein localization, and promote a binding affinity of proteins. Plays a regulatory role in gene transcription, signal transduction, and other cellular processes. Methylation of its tones has been shown to induce gene activation of genes or gene repression depending on the amino acid residue that is methylated. Methylation of histones have been shown to induce gene activity or gene repression depending on the amino acid residue that is methylated. Protein methylation has been extensively studied in relation to cancer, in which cell cycle regulation and the regulation of cellular processes go erroneously out of the right course. Then next, phosphorylation. This is the reversible addition of phosphoryl or phosphate group usually to the threonine, tyrosine or serine residues of the peptide gene. It is the commonest and one of the most important post-translational modification process in amino acids. Phosphorylation and dephosphorylation can switch on or switch off many enzymes. Phosphorylation is catalyzed by kinases and dephosphorylation by phosphatases. Protein phosphorylation can transform an uncharged protein to a negatively charged and hydrophilic protein, inducing conformational changes in it. Phosphorylation regulates the biological function of most proteins. It is very significant in signal transduction, cell growth, cell cycle, and programmed cell death or apoptosis. Then next, sulfation. Sulfation is the addition of sulfate group usually to the tyrosine residues or of peptide genes. It is accomplished by tyrosyl protein sulfotransferase, which are membrane bound enzyme trans Golgi bodies. The universal donor of phosphate group acetylation process. Protein acetylation is the addition of acetyl group to the N terminal of the peptide gene. catalyzed by the enzyme acetylase or acetyl transferase. The acetyl group donor for this reaction is acetyl-CoA. The acetyl group donor of this reaction is acetyl-CoA. Then acetylation is significant in protein stability, apoptosis, cancer, etc. Acetylation and deacetylation of histone is very important in gene regulation. Acetylation of histone reduces its positive charge. This in turn reduces its interaction with the negatively charged phosphate group of DNA and thereby makes it less tightly bound to DNA. Thus, the DNA is readily accessible to gene transcription. Acetylation of p53 protein is very important for its growth suppressing properties. p53 is a transcription regulating and tumor suppressing factor which regulates cell proliferation and responds to signals such as DNA damage by arresting cell cycle or inducing apoptosis. So when we try to learn about this post-translational modifications, major processes involved. Addition of chemical groups, that's a reversible process like hydroxylation, phosphorylation, acetylation, methylation. These processes are reversible. Then amino acid modification, that's an irreversible process like deamination, elimination, then cleavage process of protein which is irreversible, proteolysis for example, then addition of polypeptides, that is also a reversible process, ubiquitination, UBL protein conjugation, etc. and along with that isoprenylation, isoprene unit addition. Then addition of complex molecules, that is also a reversible process. For example, AMP addition, ADP ribosylation, glycosylation, etc. Suppose translational protein modifications or PTPM, process of PTPM, acetylation, incorporation of acetyl group CH3CO. Acetylation, incorporation of acetyl group, amidation, incorporation of amide group NH2 at C terminal. Then biotinylation, covalent addition of biotin. Then cysteinylation, formation of disulfide bonds between free cysteine molecules. Then deamination, removal or conversion of amide group. Then farnesylation, addition of an isoprenyl group to a cysteine residue. Then formylation, addition of formyl functional group. Then geranyl, addition of one or two 20 carbon lipophilic geranyl isoprene to cysteine. Then glutathione, addition of glutathione to cysteine. Then glycation, covalent attachment of sugar to protein. Then glycosylation, enzymatic attachment of glycan to protein. Hydroxylation, incorporation of hydroxyl group OH into an organic compound. Then methylation, addition of methyl group. Mono ADP ribosylation, addition of ADP ribose to arginine side chains. Then myristylation, covalent attachment of myristoyl group to an N-terminal glycine residue. Then oxidation, removal of electrons from a substance. Palmitylation, covalent attachment of fatty acids to cysteine, serine or threonine. Then phosphorylation, attachment of phosphoryl group. Poly-ADP ribosylation, covalent attachment of polymers of ADP ribose to protein. Then steroylation, covalent attachment of stearic acid to protein. Then sulfation, enzyme catalyzed conjugation of sulfo group. Next major process is hydroxylation. This is the addition of hydroxyl group to certain amino acids or proteins. It is catalyzed by hydroxylases. The amino acids that are usually hydroxylated include proline, lysine, asparagine, histidine, etc. Hydroxylation is important in the conversion of hydrophobic or lipophilic compounds to hydrophobic compounds. It has a significant role in the regulation of protein stability, regulation of the kinase activity of some modified proteins, etc. Then lipidation. This is the covalent bonding of lipid group or a protein. Lipidation is accomplished by different processes such as prenylation and myrstolation, palmitolation, and glycosyl phosphatidylinositol or GPI anchor addition. Prenylation is the addition of isoprenoid moiety to a cysteine residue of a substrate protein. It is critical in controlling the localization and activity of several proteins that have critical functions in biological regulation. Then myrstolation is the addition of myristoyl group to a cysteine residue, glycine residue by an amide bond. It is significant in membrane association and apoptosis. In palmitorylation, a palmitoyl group is added to a cysteine residue of a protein. The GPI anchor addition, GPI in the sense glycosyl phosphatidylinositol, GPI anchor addition, the carboxyl terminal signal peptide of a protein is split away and replaced by a GPI anchor. Recent studies in human genetics have revealed that GPI anchors are important for human health. Any defect in assembling, attachment, or remodeling of GPI anchors may lead to genetic disease known as inherited GPI deficiency. Then nitrosylation. Nitrosylation is the covalent incorporation of the nitrosyl moiety of NO into another molecule. It is physiologically important post-translational modification that affects a wide variety of proteins that are involved in a number of cellular processes. Nitrosylation taking place at the thiol group of cysteine is termed as nitrosylation. On the other hand, the nitrosylation taking place at a transition metal, the catalytic site of a metalloenzyme, is termed metal nitrosylation. S-nitrosylation is a ubiquitous regulatory mechanism for protein conformational change, protein-protein interactions, and further post-translational modifications such as phosphorylation, acetylation, ubiquitination, and sulfide bond formation, disulfide bond formation. Then ubiquitination. Ubiquitination is the addition of ubiquitin to the lysine residue of a substrate protein. Ubiquitin is a small protein, 8 kilodalton. Ubiquitination regulates a protein's function or marks it for degradation. Either a single ubiquitin molecule, monoubiquitination, or a chain of several ubiquitin molecules, polyubiquitination, may be attached to the protein. Polyubiquitinized proteins are recognized by 26S proteasome and subsequently targeted for proteolysis or degradation. Monoubiquitinated proteins may influence cell tracking and endocytosis. Then ubiquitination serves several functions. The commonest one is to flag proteins for degradation by proteasome. The other functions include immune response, inflammatory response, organelle biogenesis, and signaling roles in DNA repair. And then SUMOL YLATION, simulation. I don't know how to pronounce it, sorry for that. It's the binding of SUMO, small ubiquitin related modifier protein. Ubiquitin related modifier protein, 100 amino acid residues protein, and target proteins in the same way as ubiquitin binds with target proteins. It is a reversible PTPM in which small ubiquitin like modifier SUMO proteins are covalently attached to the lysine residues of a target protein. It confers transcription regulation activity on target proteins and helps in the transportation of target protein from cytosol to nucleus. Then AMPylation. AMPylation is the reversible addition of AMP to protein. It involves the formation of a phosphodiester bond between the hydroxyl group of a protein and the phosphate group of AMP. Later, deamination. Deamination is the removal or Conversion of asparagine or glutamate residues to another functional group. Asparagine is converted to aspartic acid or isoaspartic acid, while glutamine is converted to glutamic acid or pyroglutamic acid. This modification can change the structure, stability, and function of proteins. So we learned many small additions, subtractions, capping reactions, etc. related to the post-translational protein modification, like protein folding, chaperones and folding of polypeptides, proteolytic cleavages, chemical modification like glycosylation, protein methylation, phosphorylation, sulfation, acetylation, then some other topics like hydroxylation, lipidation, nitrosylation, ubiquitination, small ubiquitin related modifier protein addition, AMP addition, deamination or removal of asparagine or glutamine residues to another functional group. So now let's learn importance of post-translational protein modifications. They play a significant role in generating the structural heterogeneity and functional diversity of proteins, bring about the conversion of one type of proteins to different functional types in different cell types, bring about the conversion of one type of proteins to different functional types in different cell types, bring about the conversion of one type of proteins to different functional types in different cell types, play an important role in the translocation of secretory proteins and export proteins across biological membranes. Some of these proteins are destined to get incorporated with plasma membrane and the membranes of lysosomes, mitochondria, chloroplasts, etc. Aid in proper protein folding. Some lectin molecules called calnexin bind to glycosylated proteins and assist in its folding. Then confer stability on protein. Glycosylation can modify the stability of proteins by increasing protein half-life. Protect proteins against proteolytic cleavage by blocking the cleavage sites. Bring about protein sorting or protein translocation. If phosphorylated mannose residues are present in the protein always goes to lysosome. Regulate protein activity and protein functions. Phosphorylation of proteins is a reversible PTPM which activates proteins. So play a significant role in generating the structural heterogeneity and functional diversity of proteins. Bring about the conversion of one type of proteins to another functional types in different cell types. Play an important role in the translocation of secretory proteins and export proteins across biological membranes. Some of these proteins are destined to get incorporated with plasma membrane and the membranes of lysosomes, mitochondria, chloroplasts, etc. Aid in proper protein folding. Some lectin molecules called calnexin bring to glycosylated proteins and assist in its folding. Confer stability on proteins. Glycosylation can modify the stability of proteins by increasing protein half-life. Protect proteins against proteolytic cleavage by blocking the cleavage sites. Bring about protein sorting or protein translocation. If phosphorylated mannose residues are present in the protein always goes to lysosome. Regulate protein activity and protein functions. Phosphorylation of protein is a reversible PTM which activates protein. Then acetylation regulates many diverse functions such as DNA recognition, protein-protein interaction, and protein stability. Redox-dependent PTM is a revolutionary conserved key signaling system which influences many aspects of cellular homeostasis. PTMs are important components in cell signaling, significant during the conversion of prohormones to hormones. PTPM significantly increase the diversity and complexity of proteomes. So that's all about this topic. Thank you for listening. Next topic of reverse transcription will be dealt with in help of next video.